Of course, this is where we left off with the last teaching group, and we're actually ready to move on, I believe. We talk a little, as I said, in greater detail about chloroplasts later in the class period, in the class semester. The next thing is peroxisomes, and peroxisomes are similar to lysosomes, only maybe you can think of them as more specialized. They have metabolic compartments that are that have a single membrane, and the compartments are such so it has a single membrane around it, and the compartments are such that what a peroxisome can do is it can produce hydrogen peroxide, which is H2O2. And so in producing hydrogen peroxide, it can neutralize things or change things that would not have been changed by the lysosomes, or actually is it's dangerous to change it inside the cell structure itself. You know what hydrogen peroxide is. Sometimes you pour it on a cut, etc., or you pour it on something to kill bacterium. And so the design of the peroxisomes is such that hydrogen peroxide can then be turned into water, so H2O instead of H2O2, H2O, and then released from the cell. So the specialized design of peroxisomes and its metabolic function allows hydrogen peroxide to be created, but then also allows that hydrogen peroxide to be converted to water. Oxygen then is used to break down different types of molecules such as fatty acid, and then also for the fuels. It goes into your Krebs cycle. Moving on to the cytoskeleton. So we're done with, I guess you could say, the membrane bound organelles. What we're going to talk about now is the area. You have your plasma membrane, but you also have an area inside the fluid environment, the cytosol, which we call the cytoskeleton. The cell itself can change its shape, but generally its shape is predetermined, and the thing that predetermines its shape is this cytoskeleton. And it is aptly named. It's kind of like, you can think of the skeletal structure of your body. It gives support to the cell, shape to the cell, and also creates basically creates these roads that things can move on. So I implied that things just float around in there. They really don't. I mean, there is some floating going on, but they also move in a very, very organized fashion, and that's because of the help of what happens in the cytosol. Three types of elements, or three different things, make up the cytosol. You have these things called microtubules, microfilaments, and intermediate filaments. Microtubules are the largest, intermediate filaments in the middle, and then microfilaments are the smallest. And this image here is showing you where mostly you see the cytoskeleton. So it's on the inner portion of the cell membrane, but before you get to the endomembrane system, and before you get to very many of the membrane-bound organelles. As I said, the idea behind these particularly the microtubules, is to create this pathway, this motorway that vesicles can walk along. And you have a great microscopic image of it, but you have, I think, probably the one that is easier to understand is the artist's rendition. So microtubules of the cytoskeleton, so this is the area that's basically right inside the cell, create these pathways that the vesicles that have been made by the Golgi, the endoplasmic reticulum to the Golgi apparatus, can actually walk along, and walk is in parentheses, but there are 2 feet type arrangements, and they do pick up and move along with the help of ATP to a spot that they are needed in the cell if need be, or to a spot where then they can fuse with the cell membrane, the plasma membrane, and be exported outside of the cell in a process called exocytosis. They're called monorails, and so you can think of them kind of as a monorails for transport. And these microtubules are so designed so that they can be recreated where needed. So they aren't just willy-nilly throughout the cell, and it's a matter of whether there's a monorail needed there for that vesicle to move. They actually can be recreated because of the design of the microtubules. Here's a nice chart. This comes from your book that is comparing sizes. So you see this microtubule to the left, just the artist's picture is bigger. The microfilaments, which are actually actin filaments, we call them actin because when we talk about we're saying that now because when we talk about muscle, their actin is one part of muscle, myosin's the other one. You have thin and thick filaments, and then the intermediate filaments, the third over in terms of size. Microtubules are hollow. Actin are two strands intertwined, and the intermediate filaments are fibrous proteins coiled into cables, which makes them very, very strong. The cytoskeleton is responsible for the ability to withstand tension. So cytoskeleton is ability to maintain shape, strength and resistance with regard to the cytoskeleton. You also have this capacity for particularly the actin, because they have the ability to withstand pressure or pull. So it's a matter of push and pull between these two in the ability to, in the job that they do. The microtubules are compression resistant, and then you also have stretch resistant portion of these. So it says maintenance on the left under microtubules, maintenance of cell shape, compression resistant, and then a microfilament, maintenance of the shape, tension bearing. So pressing with regard to microtubules, pulling in terms of microfilaments. And then also with the intermediate filaments, tension bearing elements. Also provides some anchoring of nucleus and other organelles, and that's the intermediate filament. Microfilaments, as I said, the muscle cell is designed from actin, and then with regard to microtubules, you have the design of what becomes a centriole. A centriole is what's going to lead the chromosomes to their proper portion of the replicating cells. So you have a full complement of chromosomes that are part of a human, and that full complement duplicates itself, but you have to make sure that the right set goes to each of the new cells, and that's the job of the centriole. And centrioles are designed in this microtubule design. This actual chart really puts it all together for you. And as I said, centrioles and centrosomes. Centrosome is the site of centrioles production. It's near the nucleus. It grows these microtubules, and centrioles have a design. It's called this 9-triplet design, and that design allows it to actually grow in length in a quicker way. But centrioles are the are needed for cell division. If there are no centrioles, the cell division won't proceed properly. Also, centrioles are central to the design of cilia and flagella. Cilia we find on the outside of the cell. It's used for movement, or it can be used as a singly, can also be an antenna, so it's something that receives information, as well as the flagella, which you find on the sperm cell. And here you have this the image of each of those. Actually, the difference between the two, they're both made of microtubules. The flagella's movement is different than a cilia. It's more of a movement like a fish, and a kind of a whipping movement, whereas the cilia, the movement is generally, we call it the power stroke, but you can think of it as back and forth. So a whipping movement or a fish type movement with regard to the flagella, and a back and forth in terms of the cilia. As I said, the flagella is found on the sperm cell. Cilia is found in numerous different places in the body, human body. You find it in your respiratory tract, that actually it beats say pathogens back out of the respiratory tract, so they don't get down into the lung fields. And cilia are also very, very important when they work as an antenna for the signaling of chemicals and the chemical messengers that function in the development of the embryo. This is the design of the microtubules, for basically for you to see in terms of how they grow and how they have the capacity to reassemble at will, or not really at will, but as directed, so that they are there when a vesicle needs them. Microfilaments, so those actin filaments, they're solid rods. And they are, as I said, for tension, resist pulling forces. They make up villi, and they make up the microvilli. Microvilli are found on villi in the small intestines, and they function, the actin microfilament functions with the myosin, and you see that they're bold-faced in terms of the design for a muscular or a muscle cell, and allow for that contraction of the muscle cell. And then also other motility cells. It's determined by special organelles for locomotion. So you have types of design with regard to these proteins that allow for movement. In the top left, you have what's known as the sarcomere of the muscle cell, where you have thin and thick filaments organized in a very parallel fashion. And if you go deeper into the design of the thin and thick filaments, they are able to lock onto each other and pull. So, and this all happens over and over again in a muscle cell, to pull and create the movement we think of as muscle. In the top right, you have the amoeba motion. So you have this the cytoskeleton and the proteins in the cytoskeleton that actually can extend itself out and move toward that extension, and that allows the amoeba to move away from a danger. And at the bottom, you have the ability to move gel around, so that you have this this streaming cytoplasm because of the actin filaments. And then you have the components of the cell membrane, and we're going to pick this slide up. The components and connections between cells. We're going to pick this up on the next one.